Hello. Thanks for attending my talk. My name is Brent Doiron. I'm a professor of neurobiology and statistics at the University of Chicago. I'm also the director for the Grossman Center for Quantitative Biology and Human Behavior. This is a new center devoted to theoretical neuroscience here at the University of Chicago, and we're excited to share our growth with you over the next coming years. But today I want to talk about research that was funded by the BRAIN Initiative, conducted in my lab in collaboration with several other labs, which I'll mention as we go through. And the title of my talk is Dimensions of Variability in Circuit Models of Cortex here. This is a recording, one of the earliest recordings known from cortex. It's by Lee and Jasper, published in 1953. And it's a single unit recording in cat visual cortex. And the thing that I think strikes many of us is the seemingly random and erratic state of cortical neurons here. They produce spikes at random times and with random intervals between each other. There's a fair bit of variability that's inherent in cortical response. And this has been something that's fascinated and puzzled scientists ever since this recording, really. There's two ways to think about how variability might come about in a neuronal response. The first one would be that a neuron inherits variability, or a network of neurons will inherit variability from a source that's outside of the network here. So here we're thinking of the system as a true stochastic system. The other framework would be that variability is produced internally within a network due to complex interactions, much akin to a chaotic or turbulent dynamical system here. My research group tries to navigate between these two distinct mechanisms for variability and often trying to merge them together to get a true appreciation of variability in the nervous system. And this is what today's talk is going to be about here. The data set that's going to motivate this work is a collaboration with Matt Smith at CMU, and the data was collected by Sanjeev. They were doing a delayed match to sample task in non-human primates here, where the animal had to fixate a target. One of 40 targets appeared, and then after some delay, the animal had to saccade to that target. The interesting thing about this data set is they were simultaneously recording both from visual area V4 as well as PFC during the task here. When we look at what's happening in V4, we see that where the array was positioned, the majority of the neurons were responsive to visual input on the lower left hemifield here, and this is a standard for V4. It's certainly going to encode for only one side of the visual space here. When we compare this to what's happening in PFC, even though we're only recording from one side of PFC, both sides of the visual scene are recorded. Neurons are responsive to both sides of the visual scene. We know this because PFC receives information from both left and right visual streams here. So PFC is really integrating the full visual scene. However, what I want to talk to you today is not the mean response or the tuning curves of these cells. I want to talk about the variability inherent in the system here. And the way I'm going to do this is becoming somewhat standard in the field. Here on the left, we have a covariance matrix, a spike count covariance matrix. We might have 50 or 60 neurons simultaneously recorded. We have many, many trials, and we look at the covariability across the population. We want to decompose this spike count covariance matrix into a shared component that describes the joint variability of neurons in the system, and then an independent component. And we're really interested in what's the effective dimension of shared covariance. How many latent variables or eigenvectors do we need to have to describe this up to some level of tolerance here? And a lot of this analysis, or all the analysis and modeling you'll see here, was done by a previous PhD student from my lab, Danielle Rager here. So here's some analysis of spike train variability in area V4 here. What Danielle did here is she used 5 eigenvectors to construct this shared covariance matrix. But even though she used 5, really the first eigenmode describes a ton of the variability. And when that's seen when we look at the eigenvalue of the first one is much, much larger than the other ones. the weight of this vector loading onto the neurons is all the same polarity. So this is really describing a framework where neurons in V4 are waxing and waning their activity in a trial-to-trial way in a coherent manner. When we plot the amount of variability explained as a function of the number of latent dimensions we need, we again see here that even just with one latent variable, we get almost 95% of the variability across our data set. So this is really saying V4 is really a one-dimensional source of variability here. When we do the exact same analysis from prefrontal cortex, we see that we need, in this particular data set, on average around 4 latent variables. So it's higher dimensional. The variability is more complex and more structured in PSC than it is in V4 here. So this poses a theoretical question for us, and this is where our modeling efforts are going to start here. A simple hypothesis for why is it that PSC seems to have higher dimensional variability than area V4 is that, well, PSC doesn't just receive inputs from V4. We know it receives input from both V4s, the one on the left and the one on the right, but it receives input from other areas of the nervous system. So then here, if each of these are providing low-dimensional sources of variability, then PSC will integrate these and then be higher dimensional just through inheritance here. And that's sort of on the stochastic system inheritance side of our continuum here. We can't discount this, but today what I want to do is explore the other possible mechanism here, namely that there is still just 2 sources of variability, a one-dimensional source from the left V4 and a one-dimensional source from the right V4. So that would be sort of 2-dimensional inputs coming into PSC, but it's the internal interactions within PSC that are able to amplify and expand the dimensionality of the inputs that it's receiving from V4. So I want to propose and explore this hypothesis in the remainder of the talk here. So let's build a model of PSC here. So I'm going to start with a relatively simple model. I'm going to have a bunch of excitatory cells, and they're going to be each modeled with a simple leak, integrate, and fire style spiking model here. So neurons integrate their inputs until there's a threshold. They produce a spike, and then they reset. This is a common model used in many, many studies. And the connection strength between neurons, I'm going to have just random structure within our network, random wiring within our network. The connection strengths are going to be fairly weak here. K is the number of presynaptic inputs that a neuron will receive, and the strength of a connection is proportional to 1 over K. So the more inputs a neuron receives, the weaker and weaker each individual input is in proportional sense. So sort of standard weak coupling theory here. And I'm going to drive this network with 2 one-dimensional inputs, one modeling the fluctuations from the left V4 and one modeling the fluctuations from the right V4. And these fluctuations are going to be one-dimensional. So they're Poisson processes with a one-dimensional fluctuating input that describes their firing rates. So they're formally doubly stochastic. And now when we look at the spike train responses in the raster plots on the left, we see that the neurons that a neuron PSC is either going to be deemed a left sensitive neuron or a right sensitive neuron, meaning which V4 is it getting its projections from. And the collection of PSC neurons that are getting inputs from the left V4, they tend to track that one-dimensional input from the left, similarly for the right. And given this very simple weakly coupled system, we can write down a theory for this network here. Y(I) is going to be the spike count response of neuron I in our PSC network here, and I'm going to take a linear theory here. So what's in the bracket is going to be all of the inputs that ith neuron is going to get. The first term in the bracket is all of the inputs that are coming feed forward from the V4 that it's being driven by. The second term inside the bracket is all the recurrent interactions within the network here, and you can see there's a YK there. So this is a recurrently coupled network here. What GI is basically going to be the gain function for that neuron. So here I'm going to take what's called a linear response theory. Each neuron is formally nonlinear in terms of its transfer function, but there's going to be an operating point that the network is sitting at, and I'm going to linearize around that operating point. of that linearization is this gain GI. It's going to be different for each and every neuron because of heterogeneity, but we can calculate what this gain is for all of the neurons in our network. If I have this real simple linear framework, we can write down mathematically what the covariance of the PFC network would be here. And it's this sort of algebra soup here on that second line of that equation here. I'll parse this out for you. The first term here is the covariability that would be due to the feed-forward connection. So that inner matrix V, that's the covariability of the V4 population. Of course, it's going through the feed-forward connection matrix, JFF, and then that gets passed through the gain of the neurons to produce output correlations. If there was no recurrence in PFC, that would be the only thing we would have, just straight feed-forward. The other two terms here are the correlations that will be due to the recurrent activity, or this feed-forward input being passed through the recurrence in the network here. And they morph and change correlations due to the wiring structure in the network here, but it's still relatively straightforward to compute what the total covariance would be here. When we test our theory versus our simulations, we get a really good match here. So these clouds of data here, the gray cloud is the covariability of left and left neurons. The black cloud, which is hidden by the gray cloud, is the covariability of the right and right neurons, and in the corner near the origin is the covariability of a right neuron with a left neuron here. That's in orange there. So our theory does a pretty good job at capturing the numerics in our simulation here. But if we take this theory and then push it through sort of a well-known theorem in linear algebra, the dimensionality or the rank of the covariance matrix CY, since it's the product of a bunch of matrices, it's going to be the rank of the minimum component in these sub matrices here. And since the V4 input coming in was two-dimensional, the left V4 was one-dimensional, the right V4 was one-dimensional, you take the sum and that's two-dimensional, we have here that the rank of the covariability in our simple PFC model cannot be larger than 2. It must be greater, it must be less than or equal to 2. That's a well-established theorem in linear algebra here. And lo and behold, when we do factor analysis on this and ask how many latent variables do we need to explain 100% of the variability, lo and behold, if we have one latent variable, we get roughly 50% of the variability. If we have two latent variables, we get 100% of the variability in our model. So this is not a good representation for what's going on here. I'll remind you that the data showed that the variability wasn't well explained by 2 dimensions, but really on average 4 dimensions here. So this simple weakly coupled model of PFC isn't going to cut it. And if we wanted to have higher dimensional activity in PFC, it would need to come from more outside sources. Our hypothesis 1 that I'm not tracking here. I want to explain it with internal dynamics in PFC. So what are we going to do here? Well, let's make the coupling between cells larger. So let's take this 1 over k coupling and turn it into what's 1 over square root k coupling. This is a standard scaling that is familiar to people that work in balanced network theory. And what balanced network theory means is that the excitatory coupling is fairly large. 1 over square root k is much, much larger than 1 over k. And because recurrent excitation is now quite impressive and quite large, it needs to be counteracted and balanced by recurrent inhibition in the system here. So we need to have excitatory and inhibitory cells to make sure that the system is stable despite this large recurrent coupling here. So now we include inhibitory cells, we give them a right and a left side projection from V4 as well. And when we do simulations for this network, we get really different dynamics. If we just do it as is here, we get bistable dynamics. So that the left neurons, which are on the top here, when they're firing vigorously, they're actively suppressing neurons that prefer the right V4, and then vice versa. You get this bistable switching dynamics that occur in this network, a really nonlinear property. This would not be captured by a simple linear system. There's really two solutions here battling it out. When we look at correlation structure, we get that neurons within the same, preferring the same V4 input, they tend to be positively correlated. Neurons that prefer opposite V4 inputs are negatively correlated, and this has been well described in papers previously from my research group. This, unfortunately, doesn't exactly look like the data. While neurons that prefer the same visual hemifield are positively correlated, that's in the blue and the cyan, that's not the case for neurons that one neuron that prefers the left and one neuron that prefers right. They're weakly anticorrelated, but not strongly anticorrelated. We can break this strong anticorrelation by now taking our network and coupling neurons in an attractor framework here, where neurons that like the same visual input tend to wire with one another with stronger connections here. So this is this clustering coefficient. So on the left, clustering coefficient of 1 would mean that there's no structure whatsoever in PFC. Clustering coefficient of 2.4 means that neurons that like the same hemifield input tend to couple with one another with a strength of 2.4 that of the opposite coupling here. So when we put this clustering into our system, we can get rid of this sort of pathologic bistability that doesn't really describe our network that well here. And that's what we see here. The system becomes less and less bistable as we put in higher and higher clustering. And again, this is work that has been explained in previous papers in my lab. When we look at the dimensionality, this is new though, no one's seen this. When we look at the dimensionality of our network, when our clustering is when there's no clustering in our network, the dimensionality in the model is still quite low. That's the C=1 solution. As we put clustering in the model, the overall dimensionality that we would pick up from factor analysis grows and starts to match what we saw in the data here reported. So this is somewhat satisfying. And putting attractor-like networks in PFC is relatively standard now. We do think of PFC as a network that is tuned for specific inputs. So let's go a little farther with this. So now that we have a model that replicates the data in PFC reasonably well, let's sort of open the hood of the car here and understand a little bit more. What we can do is we can take the dynamics of our model and segregate periods of time when the model is such that the left network is dominant over the right, that's zones on the raster plot, or periods of time where the right is dominant over the left, that is in the orange periods of time. So we can segregate our network into being into one of two states, and these intermediate transition zones, we're going to throw them out of our data set right now and not analyze them here. So when we restrict our network to being in one of the two states, in this particular case, the left state, so that means neurons that prefer the left input are highly active, and we do our linear theory where we estimate the gains and we take our linear response functions, we do a pretty decent job at explaining the covariability in the network. On the y-axis is the theory we would get from fitting the gains and using that matrix formula there versus what we measure from the simulations on the x-axis here. And does a pretty decent job at explaining everything, despite the fact that the coupling is fairly strong here. Remember, this is the strongly coupled network. Interactions are quite impressive here. But if we restrict our system to being in the state L here, we get a pretty decent job. If we restrict our system to being in the state where the right PFC neurons are dominant over the left, we also get a pretty decent match between theory and sense. However, if we look at the gain function, we see that if we linearize our system where the neurons are in the left state versus the right state, the gain does change here. So those GIs are different depending on which state we are in. That's again a hallmark of a nonlinear system. So the gain or the slope of the transfer function of a neuron changes depending upon which state it's in effect sampling the nonlinearity of that neuron's transfer function. And that's true for all of our neurons here. So here, this is sort of the last couple slides here. If we do our dimensionality analysis and we ask what's the cumulative variance explained, shared variance explained, how many dimensions do we need to explain 95, 98, 99% of the variance? if we restrict our system to being in one of the two states, either state R or state L, it looks like we just need two dimensions. And that makes sense from our linear theory before. There's two V4 inputs, they're both one-dimensional. PFC is receiving both of them. If it's acting like a linear system, then it'll just inherit the dimensionality of those V4 inputs, and you get the answer 2. However, if we look at the system for the full dynamics, where we're allowing the transitions to happen, now in order to explain a high percentage of the variability, we need a higher dimension. We need three or more dimensions here. And this is because these two-dimensional manifolds given by the linearizations are different depending on whether you're in state R or state L. And the network is transitioning between these. And if you build a linear theory that stitches these two things together, you can do a better job at this. And this is effectively saying, well, when you look at the full system, there's two-dimensional variability when the system's in state R. There's another manifold, which is two-dimensional when the system is in state L, and sort of the union of these two manifolds is effectively 4 dimensions here. This is sort of not a proof in any sense, but sort of a good way to think about why variability, the dimension of variability, is expanding when we go from V4 to PFC. It's the nonlinearities in PFC that sort of sample different regions of activity space here and amplify the inputs here. So some of the takeaways here from this is that visual area V4 has very low dimensionality, while PFC shows a higher dimensionality. This expansion from V4 to PFC in terms of dimensionality can be captured if the PFC networks are strongly coupled and they have a tractor structure with their connectivity. And we can get a good understanding of this dimensionality expansion by stitching together linearizations of the network about distinct states here. So this is the beginning of a path that I'm taking with my lab and my collaborators to understand how the dimensions of activity are transmitted across cortical networks. As we start recording from more and more regions of the brain simultaneously, these kinds of theories will be more and more important to see here. My last slide is to give acknowledgments here. So really the work, majority of the analysis and modeling work was all done by Danielle Reger, who was a graduate student in my lab. Now she works for IBM in their neuromorphic chip division. And this is a collaboration with Matt Smith as well as Byron Yu. And it really comes out from a Brain Initiative Award from the Theories, Models, and Methods for Analysis of Complex Data from the Brain. This has been a great mechanism for me to receive my lab and my collaborators to receive funding. So I thank you for that support.